So in continuation with the genetics topic, in the previous video we have discussed about the introduction and brief account on Mendel's work. Now we'll go through Mendel and his experiments. As we know, Mendel selected pea plant, that is Pisum sativum, for his experiment. And why Mendel selected pea plant for his experiment? The question arises. Sometime it will be asked for 2 marks. Why Mendel selected garden pea plant for his experiments? Why not the other plants? Why not the other animals he has not selected? Because of the following advantages, Mendel selected garden pea plant for his experiments. So the advantages are: first one, it is a short day annual plant. Short day annual plant means completes life cycle within a season, within a year, so that we will get more and more number of generations within one season or within 1 year. Second advantage of selecting the pea plant is it has perfect bisexual flowers and they are self-pollinating. The plant has got perfect bisexual flowers and they are self-pollinating. Due to self-pollinating or due to self-pollination, the plants are homozygous. Homo means similar. Individual or the zygote having similar genes for a character. So they are homozygous. So it is easy to get pure lines for many generations. Pure lines means having the same character without any change in generation to generation. Pure lines. And those flowers are easy for crossing also. Because of these advantages, Mendel selected garden pea plant for his experiment, for his experimental work. So after selecting the garden pea plant for his experiments, and he recognized 7 pairs of contrasting characters which were related as dominant and recessive. He identified or he recognized 7 pairs of contrasting characters which were related as the dominant and recessive. Those 7 pairs of contrasting characters are: first one, length of the plant, in which tall is dominant over short or dwarf. Tall is dominant over short or dwarf. Second character is position of flowers, where the dominant is axial, recessive is terminal. Dominant is axial, recessive is terminal. And third character is pod shape, in which dominant is inflated, recessive is constricted. Dominant is inflated, recessive is constricted. Then pod color, dominant is green, recessive is yellow. Then color of seed coat, gray is dominant, white is recessive. Shape of the seed, round is dominant, wrinkled is recessive. And color of the seed, that is yellow is dominant, green is recessive. Like that, he recognized 7 pairs of contrasting characters in garden pea plant, that is Pisum sativum. And after recognizing and after selecting and after recognizing the garden pea plant for his experiments, Mendel started his work. The work of Mendel is monohybrid cross. The work of Mendel is, first one is monohybrid cross. So what is monohybrid cross now? The cross which involves only one pair of contrasting characters or 2 alleles of a character is called monohybrid cross. The cross which involves only one pair of contrasting characters or 2 alleles of a character is called monohybrid cross. So let us consider the character from plants, that is in pea plant. The character first Mendel considered is length, in which dominant is tall, recessive is short or dwarf. Dominant is tall, recessive is short or dwarf. In genetics, we will represent the characters by the first letter of dominant trait. For tallness, capital T, and its second alphabet, that is small t for shortness. So in monohybrid cross, Mendel selected only one pair of contrasting characters, that is tall and short. He crossed homozygous tall pea plants with homozygous short pea plants. So because of their homozygous condition, both produces only one type of gametes. This is parental generation, represented by P or P1, and this is genotype of the parent, and this is gamete of the parents. By crossing these 2 gametes, we will get the first generation with the genotype capital T, small t. This is called F1 generation, that is first filial generation. By crossing homozygous tall pea plants with homozygous short pea plants, in F1 generation, Mendel got all the pea plants were tall, but they are heterozygous. Were tall, but they are heterozygous. And for the next generation, that is for F2, Mendel allowed F1 plants for selfing. F1 is a selfed. So F1 selfed means heterozygous tall pea plants with heterozygous tall pea plants. Because of their heterozygous condition, they will produce us 2 types of gametes. That is 50% of the gametes with tall genes, 50% of the gametes with short genes. 50% of the gametes with tall genes, 50% of the gametes with short genes. By crossing them, capital T with capital T, capital T with small t, small t with capital T, small t with small t. This is F2 generation. This is F2 generation. So in F2 generation, Mendel got 25% of the plants were homozygous tall. 50% of the plants were heterozygous tall. And 25% of the plants were homozygous short. Homozygous short. So the ratio, genotypically it is 1:2:1. This is genotypic ratio. Phenotypically the ratio is 3:1. Phenotypic ratio. Genotypically 1:2:1 and phenotypically it is 3:1. Genotypically means which can be written by considering the genotypes of the individuals, and phenotypically it is just by observing. For example, in 100 plants, if 75% of the plants were tall, then it is 3%. If 25% of the plants were short, that is 1, 3:1, like that. I think you will understand with this monohybrid cross example. Go through the notes that I am going to give you in the form of PDF. If you have any doubts, any queries, or any clarification about up to the monohybrid cross, you may contact me or you may call me, either message me. I will respond to the questions or queries of you. Thank you.